The topic of gene therapy has been discussed for generations at this point, and it's really coming to a head, if you will, in retina. There's a tremendous number of gene therapy programs, and that's such a broad term. So this data in particular is looking at creating an intraocular biofactory that produces an anti-VEGF agent that can be used for wet AMD, a very common cause of blindness globally. There are other gene therapy programs looking at very small numbers of patients with inherited retinal diseases, and then there's the spectrum in between of all sorts of different populations of patients being studied for gene therapy. Geographic atrophy, another one, very large population of patients. Diabetic retinopathy, another one, being studied gene therapy. So it's important to pretty quickly with gene therapy dive into what population of patients we're looking for. But if you stay at the high level, the benefit of gene therapy, the concept is that you can potentially have a one and done. And I use that word potentially very importantly because there's no guarantee yet in any of these programs that you're going to treat a patient and they'll never need another treatment for that disease. But that's the goal, is to treat someone once and create an intraocular biofactory, which basically means you're taking the cellular machinery in the eye and you're using that to produce something different than that cell has produced in the past. Maybe you're producing something like RPE65, which is replacing a gene that is not working in that cell type that should have been working in the ideal situation. Or maybe you're producing an exogenous protein like an anti-VEGF agent, like aflibercept in the context of Ixo-vec. And so the devil's in the details with gene therapy, but the hope is that you can do a one-time therapy and then meaningfully change the course of disease going forward for whatever that disease is. So if you hone that lens down then and just look at wet AMD, for example, not thinking about dry AMD, that's a different topic, but wet AMD, there's 3 major gene therapy programs in advanced or sort of middle stage human trials. REGENXBIO has a very important pair of phase 3 trials, ASCENT and Atmosphere, which is a subretinal, so it's transvitrectomy. So you're in the operating room, you do a vitrectomy surgery, and then you inject RGX-314, which produces ranibizumab, under the retina, and you're transfecting the RPE cells to turn them into a biofactory to produce ranibizumab. And they've had good data to date, not from the pivotal trials yet, but from earlier stage trials, and they have 2 global ongoing trials right now that will help us better understand that target product profile, and I'm hopeful that those will be successful and be commercially available in the future as the first gene therapy for wet AMD. And then the other 2 major programs are one led by Adverum and one led by 4DMT, which are intravitreal viruses that are being used to produce aflibercept to also treat wet AMD. And the value proposition there is that you don't need to go to the operating room. It's an intravitreal injection. And when you do intravitreal injections, there's risk there that we've seen with these programs of inflammation, which we have not really seen with the RGX-314 program subretinally. The other angle to add for wet AMD, of course, is RGX-314 has been used suprachoroidally. So that virus has been given subretinally and also suprachoroidally, and that's at an early stage of development. So all of those are promising programs. They each have their potential benefits, their challenges, and they're all continuing to move forward.